And the other equally important event that needs to occur is that the cancer cell needs to inhibit tumor suppressors, like the retinoblastoma protein and the tumor suppressor protein P53, which can direct cell cycle arrest, senescence, or apoptosis of damaged cells. The tumor suppressor P53 is perhaps one of the most important tumor suppressors described to date. It is mutated in over 50% of all human cancers. And in those cancers where it is not mutated or altered, there is some kind of alteration that affects P53 function in one way or another. P53 can be activated by a variety of different stimuli, including ER stress, oxidative stress, oncogene activation, hypoxia, ionizing radiation, UV, DNA damage, or telomere erosion, attrition. All these different kinds of stimuli activate and stabilize P53, which can then direct metabolic homeostasis, antioxidant defense, DNA repair, growth arrest, senescence, or apoptosis, depending on the severity of the damage and whether the damage can be repaired or if the damage is so severe that it requires death of the cell in order to maintain tissue homeostasis. P53 is a member of the P53 family, which includes P53, P63, and P73. These last two were discovered only about 15 years ago, less than 15 years ago, based primarily over sequence homology over their transactivation, DNA binding, and oligomerization domains. All three genes are transcribed as different isoforms that differ in their N and C termini. In the case of P63, half of the isoforms contain a transactivation domain that is about 22% homologous to the transactivation domain of P53, while the other isoforms, the delta N P63 isoforms, are transcribed from an intronic promoter that is spliced out in TAP63 isoforms and thus bears no resemblance to P53 on the N-terminal transactivation domain. Additionally, alternative splicing generates 5 different C termini, alpha, beta, gamma, delta, and epsilon, for a total of 10 isoforms. The DNA binding domain and the oligomerization domain are shared by all isoforms and are 60% and 37% homologous to those of P53. The alpha and the beta isoforms contain a second transactivation domain, and the alpha isoforms contain a sterile alpha motif, which is important for protein-protein interaction, and a post-inhibitory domain, which is important for intramolecular inhibition of gene transactivation on the TAP63 isoforms. From the very beginning, it was clear that P63 is very important for development because P63 mutations are strongly associated with human developmental diseases, which are generally characterized by ectrodactyly, cleft lip and palate, ectodermal dysplasia, and a few other developmental symptoms. And what's very interesting is that specific syndromes, like the EEC syndrome or the AEC syndrome, are associated with specific mutations in specific domains of the protein. So for example, EEC, which is ectrodactyly ectodermal dysplasia cleft lip palate syndrome, is associated with these mutations localized on the DNA binding domain, while ankyloblepharon ectrodactyly cleft lip palate syndrome is associated with these mutations on the SAM domain. And I just want to bring your attention to these two mutations here, cysteine 306 to arginine and 16526 to tryptophan mutations, because I will refer to these later as I use them in some of my experiments. These phenotypes are generally more or less recapitulated in the P63 null mice, which exhibit early postnatal lethality due to a complete absence of all stratified epithelium, so no skin whatsoever, and ectodermal appendages such as the hair, mammary glands, the teeth, the nails, in addition to truncated limb development and severe craniofacial defects. So it is more than clear that P63 is absolutely essential for proper development. But the role of p63 in cancer has been much harder to elucidate, primarily because of the presence of so many different isoforms with seemingly opposing functions. So on the one hand, TAP63 isoforms promote cell death. Exogenously, TAP63 can induce cell cycle arrest, can induce senescence and apoptosis, and can inhibit tumor growth when expressed in mouse xenografts. An endogenous TAP63 is absolutely required for DNA damage-induced apoptosis in oocytes in a manner that is independent of p53. But then, on the other hand, delta NP63 isoforms promote survival. The predominant p63 isoform is the delta NP63 alpha. It is expressed in epithelial cells and in the basal cells of stratified epithelia, like in this example of the skin, and its expression is progressively lost in the suprabasal layers. And it has been shown that this isoform, delta NP63 alpha, is absolutely required for maintaining the proliferative potential of stratified epithelial stem cells, and its ablation results in proliferative rundown. Now, because of its importance in promoting survival and maintaining stemness, it is actually not very surprising that delta NP63 alpha is usually upregulated in low-grade squamous cell carcinoma, as you can see in these biopsy examples of lung, tongue, and head and neck squamous cell carcinoma, compared to normal lung, normal tongue, and normal tonsils. But what is much more surprising is that reduced p63 enhances metastasis. In this mouse genetic study performed by Tyler Jack's group at MIT and Elsa Flores' group in Texas, these authors found that p63 haploinsufficiency in a background of p53 heterozygosity results in much increased metastatic frequency. This is a very important finding because it suggests that p63 could be a metastasis inhibitor. So we said, well, okay, this is mouse genetics. What about human? Is there a correlation between cancer progression and p63 expression? So we used Oncomine to look at the expression of p63 at different stages of human cancers. And we found that as cancers become more aggressive, p63 expression is progressively reduced. So look at this metastatic prostate cancer lesion and compare it to a benign case of prostate cancer, or a metastatic melanoma compared to primary melanoma compared to a normal mole, or a grade 3 versus a grade 2 esophageal squamous cell carcinoma. So we were actually very excited. This was like big news. We had found that p63 expression decreases in advanced cancers. And we know that delta NP63 alpha is the predominant p63 isoform in epithelial cells. So we asked, could delta NP63 alpha, the predominant isoform, be a metastasis inhibitor? To begin answering this question, Johann Wu from our lab overexpressed delta NP63 alpha, which from now on I'm just going to refer to delta NP63 alpha as p63, in HS578D breast cancer cells. These cells don't express p63 that can be detected by Western blotting and are highly invasive but not tumorigenic. Johann also overexpressed two mutant derivatives, one defective in DNA binding and the other one defective in protein-protein interaction via the SAM domain. And then he subjected these stable cells to cell invasion assays and found that only those cells that expressed wild-type p53 and not those cells that expressed mutant derivatives of p63 had reduced cell invasion. This was very exciting again because it supports the notion that delta NP63 alpha could be a metastasis inhibitor, and it points out to the importance of direct DNA binding and of protein-protein interaction via the SAM domain. So then we asked the question, well, what could be some of the molecular mechanisms responsible for mediating these effects? And to answer that question, we profiled gene expression using Affymetrix technology, and I decided to focus on those genes that were upregulated by wild-type p63 but not by the mutant derivatives. So I took these genes and I classified them onto